Hi, my name is Vencic. I'm a PhD student at the University of Sherbrooke, Quebec, and you are listening to the recorded presentation of my scientific poster for GLBIO2023 entitled Using Machine Learning on Single-Cell RNAseq and Clinical Data to Disentangle Response to Treatment Uncertainty and Rheumatoid Arthritis. Begin with some background on rheumatoid arthritis. Rheumatoid arthritis, or RA, is a chronic inflammatory and autoimmune disease that causes joint pain, swelling, and irreversible damage. This inflammatory type of arthritis affects about 1% of the adult population worldwide. It is characterized by an alternation of asymptomatic phases and symptomatic flares. However, the physiopathology of RA is still not clearly understood, and there is currently no cure for RA, with available treatments only inducing temporary remission when effective. RA is characterized by specific symptoms such as joint inflammation, progressive destruction of joints, symptomatic flares. Those symptoms will lead to a diagnosis of rheumatoid arthritis. Over several patients diagnosed with comparable profiles, we would expect consistency in treatment response. Surprisingly, we observe variability in treatment response, that is, several patients not responding to the same treatment and even some patients not responding to any available treatments. This led us to posit that this difference is due to a plurality of immune dysfunction causing specific symptoms from distinct immune cell expression profiles, namely the RA endophenotypes. This leads us to our scientific question: how to characterize distinct immune endophenotypes and investigate correlation with RA clinical outcomes. We decided to conduct this study using single-cell RNA sequencing. Here, we can see the data obtained with classical or bulk RNAseq, which give us the expression for each gene in a given sample. But one sample is often constituted of several cell types with their distinct role and expression profile. This is where single-cell RNA-seqing is interesting, because it allows us to observe the expression level of each gene in each cell present in the sample. This is crucial to complex sample composition, such as our project with circulating immune cells, which are highly specialized. To analyze the PBFC samples collected from patients, we'll build a single-cell RNA-seq pipeline composed of several standard steps. First, we map the reads to a reference genome. We continue by filtering out the low-quality genes and cells to reduce data noise. Then, we cluster cells sharing a similar expression profile. We finish by automatically annotate those cell clusters with cell typists by computing the genes specific to each cluster and then interrogating marker genes databases. Running the previous pipeline over our dataset composed, as of now, of 9 newly RA diagnosed patients and their 9 matched control, we obtain this UMAP, where each point represents a cell and its position is tied to its global expression profile relatively to the other cells, such as cells sharing a similar profile are close. The automated annotation tool CellTypist outputs 16 immune cell subpopulation for our dataset. Integrating the currently available treatment information of 4 patients, we investigated the difference in cell subpopulation distribution among treatment response. Taking as example classical monocytes, we then investigated further down the transcriptomic characteristics of each treatment outcomes by analyzing the intersection of differentially expressed genes between classical monocyte cells of each patient and its matched control, showing specific genes to each treatment outcome. Then need to be able to investigate expression profile differences among patients in cells from a specific subpopulation regarding treatment response. To do so, we start by averaging the expression of each gene over all cells from the same cell type in each sample. Afterward, we reduce dimensionality applying PCA and UMAP to the mean gene expressions. This gives us For each sample, reduce variables for each cell subpopulation, reflecting its expression profile. Applying this process to the classical monocyte in our datasets, we observe that samples with different treatment response are distant from each other. This study is awaiting more data to refine our methodology and validate our preliminary findings. The long-term objective being to build a machine learning-based tool able to predict area treatment response and course of newly diagnosed patients. This ends the record. Thank you very much for listening, and I'll be happy to respond to your question during the poster session. Have a nice day.